A woman has a 1 in 8 chance of developing breast cancer over her lifetime. An important risk factor is age, but there are also known genetic risks.
For example, mutations in specific genes, such as the familial breast cancer susceptibility gene BRCA2, which really dramatically increases the risk for developing breast cancer.
Breast cancer is notable for inherited mutations in at least 10 genes important for genomic integrity, including BRCA2.
So it is critically important to fully understand the mechanism that cause breast cancer and the proteins that are involved. In the case of BRCA2, we actually thought that we had a pretty good idea of its cellular function. That is, that it loads the recombinase, RAD51, onto double-strand breaks to repair the broken chromosomes, and therefore prevent chromosomal instability, which is associated with tumorigenesis.
By loading RAD51 onto breaks, BRCA2 acts like a nurse, healing breaks and chromosomes. We thought that BRCA2 would have the same function during DNA replication. That is, when replication forks stall, the DNA would break and need to be repaired in order to restore replication.
But that's not what we saw. In fact, we could not detect any defect in replication restart, which is what one would expect given this broken fork model.
Instead, we found that BRCA2 protects nascent strands at replication forks. In its absence, nucleolytic degradation occurs, which is associated with chromosomal instability.
So in this case, BRCA2 acts really more like a superhero in protecting from DNA damage in the first place, as opposed to rather healing it after the damage has already occurred.
In particular, the C-terminal domain of BRCA2, which stabilizes RAD51 filaments, turned out to be essential for fork protection.
On the other hand, we show that RAD51 filament stabilization is actually not important during double-strand break repair. So we really are looking at something quite different here mechanistically at those stalled forks, where BRCA2 somewhat converts RAD51 into a structural or architectural protein, as opposed to an enzymatic one during double-strand break repair, where RAD51 catalyzes the search for homologous sequences.
We believe that RAD51 filament stabilization is important at stalled forks because physical forces oppose the open fork structure.
Nascent strands at unprotected forks are degraded by MR11 nuclease. And that is actually quite interesting because it's an example where a good protein can turn bad, or a superhero can turn into a supervillain.
In the wild-type situation, MR11 promotes error-free repair. But when BRCA2 is defective, MR11 becomes mutagenic, promoting chromosomal aberrations.
In fact, we found that we can largely reduce this type of instability by chemical inhibition of MR11.
The critical site for fork protection in the BRCA2 C-terminus is highly conserved throughout vertebrates and is even found in invertebrates. RAD51 filament stabilization is therefore likely to be critically important throughout diverse organisms, and mechanistically may occur even in bacteria.
So this really stresses the importance of this region in BRCA2, since the most common disease-causing mutations are truncation mutations, which again at a minimum involve a C-terminal deletion of the protein. Now, although this is only a hypothesis at this point, of course, there nevertheless seems to be a very strong correlation between tumor suppression and this new mechanism.
Moreover, BRCA2 reversion, which leads to therapy resistance, can restore this function to the advantage of the now tumor cell.
Since this is different than double-strand break repair, we really anticipate that this is likely not only BRCA2, but that there are other tumor suppressors involved with this function, which we think is especially important to any kind of replicating or cycling cell, such as those stimulated for growth by hormones, just to name one example. But one is certainly tempted to speculate because one can easily imagine other cellular scenarios in which this protective replicative mechanism could become very important, such as for the hematopoietic system or during stem cell maintenance.
Ultimately, it is our hope that this study with BRCA2 can serve as a starting point to understand this new function and how it can be used to improve cancer therapies and disease prevention.